Hi, class. Welcome back. It's Dr. Lindner. We're still on neurophysiology. We're going to be talking about what happens at the synapses. And some of this should sound a little bit familiar because if you think back to the introduction to muscles, we did talk about the neuromuscular junction. And we spoke about exactly this, what happens when the action potential gets to the very, very end, where you have the synaptic terminal or the synaptic end bulb. And what we have stored in that synaptic terminal are a bunch of these synaptic vesicles. And what we have in these synaptic vesicles are neurotransmitters. And remember, we need calcium that influxes in, and that's going to tell the synaptic vesicles to open up and release their contents into the synaptic cleft. That method is called exocytosis. If it's the neuromuscular junction, remember that neurotransmitter is acetylcholine, but this could be dopamine, or this could be serotonin, or this could be GABA. This could be any neurotransmitter, okay? The one that we're familiar with so far is acetylcholine, but there are others that we will learn about shortly. When that neurotransmitter crosses the synaptic cleft, it's going to bind to the channels on the post membrane, the postsynaptic side. And when those rush in, it creates the action potential, right? It's going to relay the action potential here to this other neuron. Okay, but it's going to create more of a chemical transmission using the neurotransmitters to be that relay. Okay, so let's take a look at this video.
Explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. Synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter.
Hi, this is Mark Gordon at Sanford Promise in Newtown, Connecticut.
Okay, so that was an important concept, right? That was a pretty good review of what we just said. So the action potential, or the nerve impulse, is traveling down the presynaptic neuron. It gets to the very end where we have the synaptic end bulb. We've got calcium that rushes in, and that's what tells these synaptic vesicles that contain a neurotransmitter to move to the end of the cell membrane wall and release it through exocytosis into the synaptic cleft. Now that neurotransmitter is going to bind to the neurotransmitter receptor. And when it binds, it opens up and allows sodium to rush in. And when sodium rushes in, it creates a postsynaptic potential. And what you heard on the video was sometimes it creates an excitatory message or sometimes an inhibitory message. Okay, that kind of depends on the neurotransmitters and the receptors. So the effect of the neurotransmitter can be excitatory or it can be inhibitory. A depolarizing postsynaptic potential is called an excitatory postsynaptic potential. A depolarizing postsynaptic potential is called an excitatory postsynaptic potential, or an EPSP. That's going to result from the opening of the sodium ligand channels, which means the postsynaptic cell is more likely to reach threshold, which is the minus 55, and then boom, it depolarizes. An inhibitory postsynaptic potential is called an IPSP, an inhibitory. I is for inhibitory, E is for excitatory. So an inhibitory postsynaptic potential is going to result from an opening of the potassium channels. And it's going to cause the postsynaptic cell to become more negative or becomes hyperpolarized. So you're moving further away from resting membrane potential and further away from threshold. Okay, so that means that the postsynaptic cell is less likely to reach threshold, which remember is minus 55. The closer to threshold, the easier it is, right? It's kind of like a person who has anger management issues, right? Everyone knows that one person that you always have to walk on eggshells. You have to be careful because you say the slightest thing and that's what triggers them and sets them off. That person is closer to threshold. The person that's calm, cool, and collected, it may take a lot to piss them off, right? It may take a lot to trigger them. That's because they're further away from threshold. So in that video, it also said, all right, we have different ways of getting rid of the neurotransmitter. So once it's released, there's the diffusion of it, which means it goes from an area of higher concentration to lesser, so it moves down the concentration gradient. Or our bodies have the ability of breaking them down. It can degrade them. So in the case of acetylcholine, our bodies make an enzyme called acetylcholine esterase that breaks it down. And our bodies can also repackage them and put them back. We call that uptake or reuptake. These are nothing other than neurotransmitter transporters. They're going to bring them back to storage. It's like a person that uses Prozac. We have some medications out there called SSRIs, selective serotonin reuptake inhibitors. So let's say in this synaptic cleft, it's serotonin, and you're happy and you're feeling good and life is wonderful, and you want to feel more of that niceness and you don't want to feel sad, then people use Prozac, which is a serotonin reuptake blocker. Instead of repackaging these and putting them back, people feel better when they're stimulating the postsynaptic side. So that's what some of the medications do. That's an SSRI. The problem with these is that long term, these can deplete B6, B9, a lot of your B vitamins. These can deplete a lot of B vitamins, and you need these B vitamins in order to convert amino acids into neurotransmitters. Without B vitamins, it makes it very difficult. So, although serotonin is good at blocking the reuptake of it, the longer people are on it, the more depleted in some of the vital nutrients that they need to produce the neurotransmitter in the first place. So yes, it'll still block, it'll still, if a person takes Prozac, whatever serotonin is left, it'll allow it to linger, but there may not be a lot of it. That's the downfall. That's why it tends to work pretty good in the beginning. And then also depends on their genetics, where some people have livers that process the medication very quickly. And they have specialized labs today that can actually look at your genetics and determine, rather than experimenting, well, I tried this drug, that one didn't work well. I tried that one, it lasted too long. Then they moved me to another one. Like if someone really needs this, they have different types of testing to determine the genetic makeup of that individual, of how quickly they can process a medication and which medication type works best for that individual. Okay. All right. Now, in terms of these neurotransmitters, which are involved in either creating an excitatory or inhibitory postsynaptic potential, we can also look at how quickly action potentials can take place. And sometimes one of the factors, besides it being myelinated or not myelinated, or the size of the diameter of those fibers, sometimes it's how many presynaptic neurons are firing on a postsynaptic. So spatial summation is when you have many, many presynaptic neurons firing on a postsynaptic neuron one time. That's spatial summation. When we look at temporal summation, this is where you have one presynaptic neuron firing over and over and over in sequence, in rapidly, one after the other after the other. So if we compare the time in milliseconds, comparing temporal summation to spatial summation, in spatial summation, this is the summation of effects of neurotransmitter released from several end bulbs onto one neuron. And when these fire, it looks like we're depolarizing somewhere around like 35 milliseconds. But over here, this is one presynaptic firing on a postsynaptic. Look how long that takes to depolarize. That one takes about 60 to 65 milliseconds. So this would be an analogy. My students that gang up on me, I'm one person, and I have many students at one time, and everyone unmutes their mic and they say, Doc, we don't want the test next week. When everyone screams that out, I hear the message loud and clear. Compared to one student saying, I don't want the test next week. 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 I don't want the test next week, right? So when you have one student firing the same message over and over, it takes longer, but there's more power in numbers, right? There's more power in numbers. So spatial summation, the S in spatial for speed, it's quicker than temporal for time. It takes more time to do that. Okay. So here you can see we have many presynaptic neurons and they're firing. So these are all presynaptic. Presynaptic 1, 2, 3, 4, 5, right? And then down. here is a postsynaptic. And you can see this is an excitatory postsynaptic potential, and excitatory postsynaptic potential. And it looks like there's only two inhibitory. So the excitatory is going to win. If there's more excitatory, then these get canceled out, and you're going to have an excitatory EPSP, an excitatory postsynaptic potential, which results from the sodium channels opening, and that creates an action potential or depolarization. Now, these neurotransmitters, their effects can be modified. When I come back, we'll talk about some of these neurotransmitters, how they're modified. We'll get into some of the names of some of the neurotransmitters and what their functions are.